Hello, my lovely anatomists and physiologists. Michele Glass here. We are finishing up our conversation on vision, and I wanted to come back to this chart that we've been working on this whole time. So when we look at vision and we talk about our sense organ, I think I would list here the retina. And, maybe you remind yourself that that's the eye. I don't think you'd need to do that. And when we talk about the specialized receptor cell, I think I would say, they are called photoreceptors. So I think it's fine to list them that way, but I think I would also list specifically rod cells and cone cells to remind myself about the difference there. And then finally, we need to fill in our afferent pathway. And so let's take some time to do that here. So when we talk about light, what we're really talking about is a particle of light. Now we see that light acts as both a wave and a particle, and we call this like individual unit of light a photon. So we're going to say photons are going to actually begin by penetrating that bulbar, or what we can call ocular conjunctiva. So remember, we mentioned that you have the palpebral conjunctiva lining the inside of the upper eyelid, and then it becomes called the ocular conjunctiva over the eyeball itself, and then palpebral lining the inferior eyelid. From here, we see that the light will penetrate the cornea. So that's that outermost portion of the fibrous tunic that is transparent, that allows light to enter. And then we can see there's actually two types of fluid in the eye. So we have what's called the aqueous humor, which is in the front or anterior chamber of the eye. And then the light will enter the pupil, which I'm going to make a note here is the hole in the iris. And then we're going to see that the light is going to be, I'm going to say, focused by the lens. And we're going to see that the light is going to penetrate through that vitreous humor. And it should be focused on the fovea of the retina. So if you're looking directly at something, those wavelengths of light should be focused at the fovea, where you get your highest acuity or your best vision, where everything else in your periphery is being detected by other parts of the retina. If you really want to see something clearly, it's best to, move your head and your eyes such that you're looking directly at that particular object. Okay, so once we hit the fovea of the retina, now let's go into the cellular level. So now let's say that the retinol in the rhodopsin of the rod cell, and because we didn't go into the exact mechanism in the cone cells, we just did the rod cells. So that's what we're going to do here. Retinol in the rhodopsin of the rod cell is converted from cis shape to trans shape. When we have that conversion, then we see an increase in sodium ion permeability for the rod cell. So then we have the signal from the rod cell sent to the bipolar cell. And then I'm doing another sort of long line here, because from the bipolar cell, the signal is sent to the retinal ganglion cells. And I did this line between the rod and the bipolar cell so I could remind us of the processing that we get at the retina with the horizontal cells. And then in between the bipolar and the retinal ganglion cells, we have the amacrine cells doing some processing there. From the ganglion cells, this of course forms our optic nerve, which is our cranial nerve 2. And from there, the signal is going to make its way to the brain stem, to the thalamus, of course, and to the visual cortex, which remember is in the occipital lobe. Okay, the last part of vision that we need to talk about is what is called accommodation. And so I am going to actually stop here and pick up with that in our final chapter 14.1 video. So stay tuned for that last one. And as always, take care of yourselves and each other.